Research, Kolkata. I welcome to you all to the second lecture of the annual Distinguished Lecture organized by our Center for Health Science and Technology. This is my proud privilege to conduct the lecture series organized as a part of the celebration of the second foundation day of our institute. In this entire series, we aim to learn from the leading lights in the field of medical biotechnology and bioinformatics. I believe all of you have already received the abstract brochure and browsed the 6 abstracts of the lectures and are eager to listen and interact with the highly distinguished scientists across the world. I request you to also browse our brochure to know more about our institute and the courses we are running. All contact information is given there and also visible on screen. Email us or call us if required. It gives me immense warmth and great pleasure to grace all of your presence today in the second lecture of the series by Professor Balaram. Thank you so much, sir, for accepting our invitation and being with us today. We are grateful to our director, Professor Ajay Kumar Ray, for his presence today. Thank you so much, sir. Live streaming of this talk is available on our YouTube channel, JISIASR. Please write your questions to the chat section of Zoom or comments section of the YouTube. Now, may I request Professor Ray to say a few words and introduce our today's speaker, Professor Balaram. Thank you so much, sir. Sir, Professor Ray, sir, please unmute yourself.
Yeah. So it gives me immense pleasure to introduce to you Professor P. Balaram, who is our speaker at the annual Distinguished Lecture Series, which our institute, the Center for Health Science and Technology, is organizing today. And Professor Balaram needs absolutely no introduction to the scientific community in India or in abroad. And he is one of the outstanding personalities in the field of, both chemical biology and biophysics. And he is one who has been contributing quite immensely over almost last 5 decades in this area, more than 5 decades. So I am I will just read a little bit about his achievements. Professor Balaram is currently the Emeritus Professor, Molecular Biophysics Unit of Indian Institute of Science, Bangalore, and DST ER of Science Chair Professor at NCBS of TIFR, Bangalore. He received his PhD degree in chemistry in 1972 from Carnegie Mellon University, USA, and after that he worked as research associate at Harvard University for another year. Professor Balaram was appointed as a lecturer in the Molecular Biophysics Unit at ISC Bangalore, and he served as the director of ISC Bangalore for a long time, from 2005 to 2014. And during this period, ISC Bangalore truly, they achieved a lot. And we have witnessed this institute coming out as one of the best, not only in our country, but in the whole world. He has also served as the editor of Current Science from 1995 to 2013. That's also for quite some time. Professor Balaram has received several awards and honors, including Padma Shri in 2002, Padma Bhushan in 2014, and the R. Bruce Merrifield Award 2021 of the American Peptide Society. He is the recipient of the World Academy of Science Prize in 1994 in chemistry. He is a fellow of INSA. Professor Balaram has authored more than 400 research articles, and the main thrust of his research is in the field of molecular biophysics and chemical biology to study the structure and function of designed and natural peptides. So Professor Balaram is like a sun, and the sun will now, glow with its own glory, and I will not spend even a second more between you and this great, son of science of India. Professor Balaram, we are indeed extremely honored at your presence. And as we discussed earlier, that we would look forward to getting your, very wise suggestions in the years to come in our journey towards excellence. Thank you, Professor Balaram.
Thank you very much, Professor Ray. Devabani, can I share my screen now?
Yes, sir. Sir, I stopped sharing, so yes.
Yeah. Shall I start?
Yes, sir. Please.
Okay. Can you see my screen?
Yes.
I'll make it full screen.
Yes, sir.
Can you see it now?
Yes, sir. Yes.
Okay. Yes. So then I'll begin. First of all, let me thank Professor Ray and Dr. Devabani for inviting me to this seminar series. I'm going to confess something right at the beginning. I'm going to talk to you generally, and I will keep the technicalities down to a minimum. What I thought I would discuss this afternoon is chemistry and biology in a general fashion in the age of the coronavirus. I'm going to tell you a little bit, an abridged history of the coronavirus, a little chemistry, a little biology, and then reflect on science and evolution. My first slide has pictured on it the foundational pillars of chemistry and biology. Mendeleev on the left and Darwin on the right. It's Mendeleev's periodic table which signifies the diversity of chemistry. And it's Darwin's ideas on species, their origin, which really tells us a great deal about diversity in biology. But we are going to talk in the midst of the coronavirus pandemic. So we might spend a moment to look at the burden of the coronavirus pandemic. A month ago, about 4 million people had died worldwide, and 1 million of these deaths were shared between the United States and India. And you can see that's an enormous burden. Also, when you look at the kind of daily cases of infection that we've had, you can see that infections have grown, fallen, grown again. So the pandemic has moved in phases from the first phase to the second phase. And in different countries, these phases have happened at different times. And one of the tasks of science, of course, is to try and understand why this is happening. He gives us very good advice. He says, keep your friends close, but your enemies closer. The coronavirus has been declared an enemy, so we should now know everything that is to be known about the coronavirus. Last year, when the lockdowns began, you would find that the coronavirus had captured everybody's imagination. Policemen in Secunderabad, auto rickshaw drivers in Chennai, everyone was decorating either their helmets or their vehicles with a spiky image of the coronavirus. In France, Italy, cakes and Easter eggs appeared with images of the coronavirus. When we locked down last March of 2020, it was the first time that most of us had been completely confined to the house for days on end. And at that time, I began to ask myself a question: who discovered the coronavirus? And so I'm going to begin with a personal digression. My investigations on the internet eventually led me to the discoverer of the coronavirus. And I wrote 2 articles, both of them in popular magazines, which now described my search for the discoverer of the coronavirus and what I found. It also turns out that after I wrote these articles and had contacted people in different parts of the world, my articles were reprinted. Usually you're very happy if your articles are reprinted, but in this case, my articles were reprinted in a very strange place. It came to feature in a newsletter in Ouray, Colorado. Ouray is a small town in the Colorado mountains. It has a total population of 1,000. I had found the pictures of the discoverer of the coronavirus in an archives in Arizona. It was at Northern Arizona University in Flagstaff. I just show you a little map to tell you that Flagstaff and Arizona are far away from one another, but not so far away as Bangalore. After I had done this investigation, I was interviewed by the archivist at the University of Northern Arizona University. She asked me many questions, and at the end of it, I made a comment. I said it is a tribute to the power of the internet that the road from Ouray to Flagstaff passes through Bangalore. It also seems to me that in some strange way, the virus has led us to its discovery by turning the world upside down. So the question really is, who discovered the coronavirus? Before we do that, we must look at the definition of a virus. And the best definition that I've found, one I liked, is in a philosophical dictionary of biology called Aristotle to Zoos, written by the English immunologist Peter Medawar. And Medawar defines the virus as such. He says, inasmuch as viruses are made known only by their causing disease or other pathological changes, the existence of benign viruses having no ill effects remains conjectural. No virus is known to do good. He says it has been well said that a virus is a piece of bad news wrapped up in protein. We're going to come to the protein. But the bad news, of course, is the nucleic acid, RNA in this case, which is the genetic material which provides the virus with instructions for its replication. But the packaging of the genetic material is not only protein, but there's also a phospholipid bilayer. And so if you look at the pictures of the coronavirus and you look at the small cross-section of the surface, you will see what is on the right side of this protein, of the slide. The spike protein projecting out of the surface The surface formed by a two-molecule thick phospholipid bilayer membrane and more proteins on the inside, which are those purple balls. At the beginning of the pandemic, we were all advised to wash our hands frequently with soap and water, and also to use sanitizers, which always contain alcohol. This is because both detergents and alcohol dissolve the phospholipid membrane and thereby serve to disintegrate the virus. William Osler, one of the founding fathers of modern medicine, more than a century ago said that soap and water and common sense are the best disinfectants. And this is a lesson that we must learn. But in the early days of the pandemic, I went back to the literature, which the internet now allows us to do, and I found this paper in 1967. I searched for it because I saw an article in the newspapers which pictured the lady shown on this slide, June Almeida, and said that she was the discoverer of the coronavirus. What they said was that she'd taken the first electron micrograph of the coronavirus, which is pictured on the center of this slide. You can see the spherical image and the spiky projections from the surface. On the left-hand side of the slide is the picture of David Tyrrell, the principal author of this paper and one of the leading virologists of his times. What Tyrrell and Almeida concluded in this paper was that the most interesting finding was that there were two respiratory viruses, 229E and B814, which are morphologically identical with avian infectious bronchitis virus. So here was a clue that there was a respiratory virus which had the kind of shape that you see under the electron micrograph, a new kind of virus. But when you read old papers, it's advisable to read them carefully because they give all details. And then there was this statement here that the viruses really were this, what I've marked and highlighted in yellow, strain 229E of Hamre and Prochnow, 1966. And then when I looked at the legend to the figure, I found that the picture that was shown was actually the picture of strain 229E. I then went to the acknowledgements, and when I went to the acknowledgements, I found the statement that Dr. D. Hamre had supplied Tyrrell and Almeida with the 229E virus. So I asked myself the question at this point, who is Dr. D. Hamre? So I went back to the literature and found this paper, Proceedings of the Society of Experimental Biology and Medicine in 1966, new virus isolated from the human respiratory tract, Dorothy Hamre and John Prochnow. Now, from the name, Dorothy, I realized I was now looking for a woman. The woman scientist, and she was the discoverer of the coronavirus. This is the first coronavirus demonstrated as a human pathogen. The paper was remarkable. What this paper established was that the nucleic acid in these viruses is RNA. She'd used 5-fluorodeoxyuridine and 5-iododeoxyuridine and shown that neither of these compounds actually inhibited the multiplication of the virus. It means it was not a DNA virus, but it was actually an RNA virus. Even more amazingly, she'd measured the size of the virus using classical filtration methods and found that it was 89 millimicrons. In today's units, this is, we would say, 89 nanometers. So if we go back to the 1967 paper, 1 year later, of David Tyrrell in the Journal of General Virology, you will see the methods that people were using. They were using organ culture to grow the virus. Nasal epithelia and trachea were dissected from human embryos of 14 to 24 weeks gestation. These are not experiments that can be easily done today, but these were remarkable experiments. And Tyrrell and Almeida concluded from their electron microscopy studies that the average diameter of the virus was 80 to 100 nanometers. And Dorothy Hamre's measurement was exactly in the middle at 89 nanometers. The coronaviruses were named in 1968 in an informal letter to the editor of Nature, which contains the names of Almeida, Tyrrell, and Hamre, and others. By 1975, when the official name was given by the virology societies, the name of Hamre had now disappeared. So I wondered what happened to her between 1968 and 1975. Why is she no longer there? And who is she? So I then did a literature search. Of course, I began with Google. And what I found in Google was many publications. These publications now span a rather wide period. But when I went to images, I did not find any picture of her. I did find people who were called Dorothy Hamre, but they were obviously different. They were younger ladies. They were not virologists or scientists. And therefore, I didn't have a picture of her. The literature search on PubMed, Google Scholar, then revealed that she was an extremely productive scientist, began a career in bacteriology, and transitioned to virology in a publishing career that spanned 3 decades. And for those of you who have forgotten your history, remember that the early 1940s were the years of the Second World War. Just preceding the Second World War was the Great Depression. Therefore, this was a very unusual time for a lady to now do a PhD degree, become a major researcher, and contribute so much to a field. In searching for Dorothy Hamre on the internet, I found a reference to Dorothy Hamre in an archives. This was in a collection which was called the Alexander Brownlee Collection at the Arizona Archives, Northern Arizona University. So I immediately wrote a letter to them, and you can see that the email is dated 29th of April, 2020. I asked them, I'm searching for a photograph of Dorothy Hamre, whose name you probably have as Dorothy Brownlee, and she's the person who really discovered the coronavirus. So at that time, Arizona was also under lockdown, and the curators wrote back to me saying that it would take them some time to find the photograph. When they first saw all the photographs in their collection, which they'd looked at, they were all wonderful photographs of national parks and monuments in the American Southwest. There were no photographs of people. But in response to my email, they looked further. And then they truly did discover her photographs. And at that point, I wrote my articles. By then I knew, because America keeps very good records, I knew where she was born and where she died. She retired and died in Ouray, Colorado. And that's where the newsletter came from eventually. So today, if I were asked, who are the discoverers of the coronavirus? These are the 3 people I would picture, Dorothy Hamre, David Tyrrell, and June Almeida, who gave us the picture which we see there. She isolated the virus, they took the electron micrograph, and we have the image of the coronavirus. David Tyrrell was a very interesting man. Towards the end of his career, and in fact his life, he wrote a book called The Cold Wars. This is not about the Cold War between the United States and the Soviet Union. This is about the fight against the common cold. Because after the Second World War, the maximum loss of industrial productivity was because of workers coming down with cold and not showing up for work. Everybody was looking for a cold vaccine. Most common colds are caused by rhinoviruses. Today, we know that about 30% of common colds are caused by coronaviruses. This was not known at that time. He did something very interesting. He took an old barracks, Second World War barracks, and converted it into a research center. Here, he got people as volunteers who had got cold, and then he got normal people also whom he attracted by saying, we will give you 10 days holiday free with all expenses paid. So what he did was to take nasal secretion from people who had colds, insert them into the noses of healthy people and see whether they got colds. And then from the collected nasal secretions, he would isolate viruses. Remarkable piece of research. And it is from this that the coronaviruses emerged. Dorothy Hamre was actually looking at medical students who used to show up at the University of Chicago to the health center suffering from upper respiratory tract infection. In 1996, Tyrrell summarized the field. He said coronaviruses cause acute mild upper respiratory infection, and in brackets he put common cold. With that, I think all interest in coronaviruses really subsided because they were minor, they were not causing major disease. But before I come to the 21st century, one question that I would like to raise right at the beginning is, are viruses living organisms? Now, if you look in the literature, you will find arguments on both sides. Reasons to exclude viruses from the tree of life, but also other people saying that viruses exchange genes across the super kingdoms of life. In fact, if you look at analyses of the human genome, you will find that they will reflect past encounters of human beings with viruses over 1 million years. Was Tyrrell correct when he dismissed the coronaviruses? No, he was not, because he could not have anticipated that in the year 2002, 2003, about half a dozen years after his review article, a major outbreak was there in southern China. And on the 28th of February of 2003, the Vietnam French hospital in Hanoi sent an urgent message to the WHO asking for help. They had identified patients who were suffering from some unknown illness. An Italian physician, Carlo Urbani, was immediately dispatched. And he's a remarkable man because within a few days he realized that this was a new infectious disease, and he persuaded the government of Vietnam to impose extraordinary quarantine in the entire country. The result of this is that the SARS outbreak was actually contained. Urbani contracted the virus and he died on the 29th of March. He already realized on his way to Bangkok that he was ill, and he had already asked people not to approach him. He had called for PPEs so that no one else would get infected, and eventually died in hospital. He's a heroic figure of the first SARS outbreak, and we must pay a tribute to him. But I'm going to digress into chemistry. We must know chemistry if we are able to understand the virus. Why should we know chemistry? The best definition of chemistry that I have seen is in an essay by Arthur Kornberg after he'd become old. Kornberg is the man who discovered the enzyme DNA polymerase. And this is one of the starting points of what we might call a revolution in molecular biology and biotechnology. Kornberg called chemistry the lingua franca of the medical and biological sciences. One of the saddest parts of Indian higher education is that in the medical sciences, so little attention is paid to chemistry, and this is also true of postgraduate courses in biotechnology. Why is chemistry important? Because chemistry is the study of matter, and everything that we see around us is matter. This slide is based on the very first chapter of Richard Feynman's famous lectures on physics at Caltech. Starts, matter is made up of atoms. Feynman asks a question. If in some cataclysm all of scientific knowledge were to be destroyed, and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? Feynman goes on to answer his question. He says, I believe it is the atomic hypothesis or the atomic fact, or whatever you wish to call it, that all things are made up of atoms, little particles that move around in perpetual motion, attracting each other when they are a little distance apart, but repelling upon being squeezed into one another. This is central to chemistry, because when atoms come close together within bonding distance, sometimes they make chemical bonds, and then they make molecules, and that's chemistry. But what distinguishes chemistry and biology are really length scales and time scales. Here I had the length scale spanning, beginning with ethane on the left and ending with the erythrocyte on the right. It's only a question of how large they are, how complex they are. Viruses, which occupy our attention today, lie right in the center between the ribosome on the left and mitochondria on the right. The ribosome is the structure for which Venki Ramakrishnan obtained the Nobel Prize for determining the ribosome structure. Mitochondria, of course, all of you know, are really where energy is produced in the cell, where oxygen that we breathe is the terminal electron acceptor in the electron transport chain. Today, we all know about oxygen because of the oxygen shortages during the second phase of the pandemic. That's where oxygen is used in the cell. Viruses lie right in between. Chemistry is important, and I can't emphasize this better than in this cartoon. There's this gentleman doing a crossword puzzle. He asks the lady sitting next to him, what's a 9-letter word for biotechnology? Very quickly answers chemistry. I might also say that if you ask the question, what is a 9-letter word for material science? You would also answer chemistry. But chemistry is sometimes difficult for beginning students. It's difficult for people in other disciplines, largely because, like mathematics, chemistry and mathematics can be set apart from physics and biology. Physics deals with forces that we experience, gravity. We can experience motion. We can see plants, animals, so we experience biology. But to experience chemistry and to understand chemistry, you need to learn a language, just like mathematics. Chemical structures are the alphabets of chemistry. And for mathematics, symbols and equations and the way they are manipulated are the alphabets and language of mathematics. But what is interesting is that in the general world, politicians in India understand the word chemistry and arithmetic very well. The Prime Minister, after the last general election, said that chemistry defeated arithmetic in the 2019 polls, because very often scientific metaphors are used by politicians in their speeches. What are the foundational pillars of chemistry? I have put 4, which I have chosen. Mendeleev, who represents diversity. Wohler represents unity, because it's Wohler who took ammonium cyanate and converted it into urea, and then wrote that excited letter to Berzelius, where he said, I have made urea without the use of kidney, man or dog. Urea was found in urine. It was a natural chemical. Ammonium cyanate was an inorganic material. And here an inorganic, inanimate material had been converted into something that was associated with life. Boltzmann, a physicist, gave us really our earliest ideas of dynamics. The idea of entropy, statistical mechanics. In fact, Boltzmann's ideas of entropy itself come from trying to understand heat. And that's when he understood that matter must be made up of atoms. And Pasteur, who more often is known as the founding father of microbiology and vaccines, but Pasteur also did the famous resolution of tartaric acid. That's what demonstrated the importance of three-dimensional structure on molecular properties and started the subject of stereochemistry. But where does all this matter come from? Obviously, all this matter on Earth has come from the stars, come from somewhere when the universe was formed. I draw your attention to this book. Jacob Bronowski, a mathematical physicist, did a BBC series in the early 1970s called The Ascent of Man, later appeared as a book. He describes this. He says, in all the stars, they're going on processes which build up the atoms one by one into more complex structures. Matter itself evolves. The word comes from Darwinian biology, but it is the word that changed physics in my lifetime. Why do I quote this? Because he uses the word evolve, and the word evolve is central to biology, but it is central to many, many human matters. I like his description of carbon, the formation of carbon, where he says that this forms when 3 helium nuclei collide at one spot within less than 1 million of a million. Every living creature has been formed with collision. What does this tell you? Without carbon, there is no organic chemistry. Without organic chemistry, itself is an extremely improbable virus. The Urbani SARS-CoV virus was found in 2003. By 2006, the new methods of cryo-electron microscopy had begun to be applied to the study of viral particles. And we began to get a much more detailed picture of how the virus is constructed by the constituent proteins and nucleic acid which form it. What is amazing on this slide is only one number, the mean particle diameter of 82 to 94 nanometers. So Dorothy Hamray's 89 nanometer measurement in 1966 has actually stood the test of time, even with the most sophisticated techniques available in 2006. So this is in fact a tribute to her skills as an experimentalist. The structure of the viral particle, the protein constituents, all of it is now known in the phenomenal burst of research which took place from 2003 till about 2015-16. But it also turns out that much of it has now been repeated from 2020 and 21 on the new coronavirus, which differs only marginally from the previous coronavirus. But when we talk about viruses, we talk a great deal about the proteins of the virus. And in order to justify the emphasis on proteins, I quote here from the high priest of molecular biology, Francis Crick, ironically, physicist. He says the main function of proteins is to act as enzymes. He goes on to say something very interesting for those of you who are interested in metabolites. It is at first sight paradoxical that it is probably easier for an organism to produce a new protein than to produce a new small molecule. Looks odd at the beginning, but it turns out that all the small molecules of biochemistry require a great deal of effort to produce them. He then concludes, there seems little point in genes doing anything else but protein synthesis. One of the most critical steps in viral infection is the fusion of the viral membrane with the host membrane. And much of our knowledge of membrane fusion processes has come from work on influenza virus, which also has a spiky projection, the hemagglutinin protein. And wonderful crystallographic work on the hemagglutinin protein has revealed the remarkable structural changes that need to take place for viral fusion to happen. This is spectacular work done by the late Don Wiley at Harvard and described in this review article, which I give you a reference to, by Steve Harrison. Coming back to COVID-19 and the SARS-CoV-2 spike protein. It's a polypeptide of 1,273 residues. Three-dimensional structure is now available in near atomic detail, even in different forms. This is what it looks like. On the right is the trimer. Three molecules of the protein form the spike that you see projecting out from the viral surface. I have just moved one of those molecules so that you can see it clearly. This is the 1,273 residue polypeptide chain sort of winding around in three-dimensional space, giving you this wonderful looking structure. You can see that some parts of the structure, especially the upper parts of the structure, appear to be disordered. They don't show you the beta sheets and helices characteristic of well-structured proteins. So there are a lot of disordered segments. But much more has been done in the last 1 year. Pictures like this have emerged, which rests on the surface of the cell, which the virus is going to infect. It attracts the virus binds to it. And those details of how the virus binds to it have also been studied by cryo-electron microscopy in remarkable work that's been done. Why are all these studies undertaken? These studies are undertaken because when we look for some kind of therapeutic or vaccine, what we want to do is to interfere with these processes. We would like to capture the coronavirus before it reaches its target cell. That's what vaccines might do. We'd like to prevent the coronavirus from binding to the target receptor. That's what therapeutics might do. Vaccines might also do that. So these detailed atomic level structures, near atomic level structures, are very valuable for design purposes. But let me come back now again to the pandemic. This is a comparison of daily cases in India and the United Kingdom between March 2020 and June 2021. You can see what it looks like. The top figure is for India, the bottom one is for the UK. You can see that in UK, their phases have come before. Their second phase came much earlier than our second phase. We had a lull. This lull between January 1st and March 20th was the time when the government announced victory over the coronavirus. In fact, public statements were made even in international meetings, the Davos meeting, for example. India was pictured as an example of a country which had overcome the coronavirus by its wonderful management. The virus proved us wrong. Why did this happen? This happened because we were arrogant enough not to learn from the science which was available at that time. These curves, which are with the dates along the x-axis, we knew in December 2020 that the United Kingdom was starting its second phase. We could have asked ourselves the question, what have we done differently from the United Kingdom, the United States, or any of the other countries which showed similar behavior? We had not done anything different, and therefore we would also get a second phase. Why would we get a second phase? That is what I want to show you on this slide. I'm not going to go into technical detail. Right at the bottom of the slide are two images. Those of you who are students, just look at them. If you don't know old stories, I will tell you what it is. It's the Trojan horse. The Trojan horse, of course, if you look at Greek mythology, is the horse which the Greeks constructed. They put soldiers inside the horse and then placed it before the fort of Troy. Because this is the story of the Helen of Troy. And then the Greek army sailed away. After the Greek army sailed away, the people of Troy came out and pulled the horse inside. And then, of course, the soldiers came out. And they opened the gates also, and the remaining soldiers came in. The Trojan horse is a way a virus actually is attracted by a cellular receptor, the angiotensin converting enzyme in this case. And then once this binding happens, the virus is welcomed into the host cell. And then, of course, replication of the virus can start. Look at the spike protein and its detailed structure. On this figure, the 1273 residue amino acids sequence is just shown on the outside. The various domains of the protein are shown and the abbreviations are expanded. NTD means the N-terminus domain. RBD means the receptor binding domain. Some things have to happen. First, the virus must bind to the receptor. It's a physical process. Interatomic interactions guide this binding. It's what I would call a process determined by the physics of interatomic interactions. The second step is a chemical step, a biochemical step. Proteolytic cleavage of the 1273 residue somewhere at the point where you see that red thing on the circle between 675 and 695. This is the prelude to infection. After that, you must have other structural changes which involve those heptide repeats, HR1 and HR2, for virus internalization. So many processes have to happen. If you have mutations, you affect these processes. How do mutations happen? They happen randomly. So the variants which everybody is talking about are products of random mutation and subsequent Darwinian natural selection. I'm going to come back to this in a moment, but I'll show you a picture. This is taken from the blog of the director of the National Institutes of Health in the United States, who highlights a paper on antibodies which bind to the spike protein. This is what vaccines generate. The antibodies can bind at many different places and interfere with all the functions of the spike protein. Sometimes mutations may happen, And I show you one mutation which I've marked here, which happens in the variants of concern. But interestingly, this mutation is at a residue, which is not involved in receptor binding. So how does this mutation affect the virus? We do not know at present. Let me digress back into biology in the last part of my presentation. First, I might ask you a question, because these are seminars on science. You might ask, what is science? Science is the study of nature. Everybody studies nature. Physicists, chemists, everyone. Biologists. The two most important journals in science, one is called Nature, the other one's called Science. One comes from the UK, the other comes from the United States. So if you go to volume 1, number 1, the first issue of Nature, 1869, November of 1869, Nature asked, for its very first issue, it asked the famous biologist Thomas Huxley to write an editorial. Huxley did not write the editorial. Instead, what he did was he translated from the German an essay written by the German poet von Goethe in the 18th century. And this is Goethe in translation. He says, nature, we are surrounded and embraced by her, powerless to separate ourselves from her and powerless to penetrate beyond. You can't find a more wonderful definition of nature. Wordsworth, of course, was a poet who always worried about nature. Darwin on natural selection, adaptation, and evolution. Natural selection and diversification of life. Darwin says there's grandeur in this view of life, with its several parts having been originally breathed by the Creator into a few forms or into one. And that while this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Everything is evolving. Animals, plants, microorganisms, viruses, they're all subject to evolutionary pressure. I gave you the pillars of chemistry. Here I illustrate 3 pillars of modern biology. 2 of these erected in the 19th century. Genetics by Gregor Mendel and evolution by variation and selection by Darwin. How are these connected? This is the chemistry of DNA as the genetic material, is probably one of the defining moments of 20th century. DNA, of course, transfers information from generation A. Mutations affect this information, and therefore information is always modified when it is transferred. But then what happens to mutants? They are selected by selection, then is
Yeah, for some technical issue, yes. Sir, would you please unmute yourself? Yes.
Yeah, it again suddenly went to mute. 1 second, we get through. Which I picked up to show you. And the other one small, so you can read it. The one by Carl Sagan. The second one, a beautiful one by Jacques Monod. In his famous book, Distinguished Minds seem unable to accept or even understand that from a source of noise, natural selection alone and unaided.
So sorry to interrupt.
Can you
Your
hear?
slides are not visible.
Slides are not visible now.
Yeah. Sir, would you please share the screen?
Can you see it?
Not yet, sir.
Can you see it now?
No. No, sir.
No. No. What can I? Can you hear me?
Sir, his screen is not shared yet. So would you please try once again? You may.
Okay. Difficult now to. Oh, yes. I think it may come now. Wait a minute. I share my.
Yeah. I think there is some technical issue. Yes, sir is back.
Hello, sir. Good afternoon. Can you please just reshare, stop share, and then reshare? Sir, we cannot hear you. Actually, you're mute.
Yeah, I'm at the point of sharing. Let me share now.
Yes, sir. We can see this. We can see it right
Yes.
now.
Yes, sir. It is coming.
You can
Yes,
see. You can
Yes.
see now?
Yes, sir. Thank you, sir.
Okay, I'll make it full screen. Hopefully you still see it.
Yes, sir. It is visible. Full screen.
Okay, my apologies. I don't know what happened. I'm just reading you a quotation on mutation, which I like. And what Monod says here is that even to accept or even to understand that from a source of noise, natural selection alone and unaided could have drawn all the music of the biosphere. In effect, natural selection operates upon the products of chance and can feed nowhere else, but it operates in a domain of very demanding conditions. And from this domain, chance is barred. It is not to chance, but to these conditions that evolution owes its generally progressive course, successive conquests, and the impression it gives of a smooth and steady unfolding. One has to read what Monod has said and try to understand it. Today, many people, even in discussing what is currently happening with the coronavirus, appear to have a problem in understanding mutation and evolution. I come back to Bronowski's book. Bronowski says in the book that among the multitude of animals which scamper, fly, burrow, and swim around us, man is the only one who is not locked into his environment. His imagination, his reason, his emotional subtlety and toughness make it possible for him not to accept the environment, but to change it. And that series of inventions by which man from age to age has remade his environment is a different kind of evolution, not biological, but cultural. I call that brilliant sequence of cultural peaks the ascent of man. This book traces the ascent of man, but you will see that every cultural peak that he talks about is built on an advance in science and technology. From the invention of fire, from the invention of stone tools, all the way to agriculture, domestication of horses, to the invention of paper and printing. All of this leads to cultural advance, but everything is based on advances in science and technology. The two dominant technologies of our times have their roots in fundamental science. Biotechnology rests on average establishment of DNA as the genetic material. Information technology rests on Claude Shannon's mathematical theory of communication, but his 1940s PhD thesis is entitled An Algebra for Theoretical Genetics. Sort of interesting today that today's information technology may make its major contribution in understanding the vast amount of biological information which is pouring into databases. Over the course of the last 20 years, so many eukaryotic genomes have been sequenced, and comparative genomics has reached a very advanced level. But what is comparative genomics is? It taught us about the unity of nature. Human beings, chimpanzees, rats, mice, pigs, cattle, sheep, horses, dogs, all treat. Sometimes when you read about the kind of conflicts that we have, you're amazed that people do not understand that biology is in fact teaching you unity. One question might be asked, what's the difference between biology and culture? If biology evolves, can culture also evolve? Is culture transmitted? Cultural transmission is more complex than genetic transmission. It can occur in short time scales, even within a single generation. It largely has to do with the way human beings think and behave. But you might ask a reverse question. Can cultures now affect genetics? There's one rather interesting example which is available in the literature. For much of human history, there was no reason for human beings to digest milk after weaning. And so once babies were weaned from their mothers, they did not drink milk again. This is largely because there is an enzyme which digests lactose, the enzyme lactase, which converts it into glucose and galactose. The lactase gene shuts off during human development or during the development of mammals in general. And once the lactase gene shuts off, you can't digest milk. And if you can't digest milk, you won't have it. But for a long period, there was no milk because dairy farming had not been invented. Humans were just like animals, hunter-gatherers. Once dairy farming was invented and dairy products became a nutritional source, people began to drink milk. What then happened? Those who could not digest it and who had the severest diarrhea then simply died because hydration was not known then. Oral hydration therapies were not known at that time. Over a period of thousands of years, human populations were selected. And once they were selected, you then had people who were lactase persistent. That means they were now what we would call mutants, in whom the lactase gene continued after infancy. Today, if you analyze the lactase gene, lactase persistence phenotype, if you go from Scandinavia all the way to the heart of Africa, you will find that Scandinavians and the Northern Europeans have lactase persistence. Most Africans do not have the lactase gene and therefore are lactose intolerant. So you can't give them too much of milk in order to make them get better nourished. If you go from north of India down to the south of India, you will find the distribution of lactase persistent varies widely. Many more people have lactase persistence in the north. Fewer people have lactose persistence in the south. And therefore, you come across many stages of lactose intolerance. This is a dramatic example of cultural evolution having influenced biological evolution. Is there a connection between history and biology? Many of you would have seen this book, Sapiens, by the Israeli historian Yuval Harari. Harari has a wonderful understanding of biology, and he writes beautifully. He says that the immense diversity of imagined realities that Sapiens invented and the resulting diversity of behavior patterns are the main components of what we call culture. Alterations of what we call history. He adds that it's independent from biology. When did the cognitive revolution happen? When human beings began to differentiate from began to develop, and this would happen along stationary societies, protecting fields now began to appear. And over a period of time, in order to create larger and larger societies, one needed to account for ideas, images, and fantasies. Harari says that to understand the rise of Christianity or the French Revolution, it is not enough to comprehend the interactions of genes, hormones, and organisms. It is necessary to take into account the interaction of ideas, images, and fantasies as well. Today, in many of the conflicts that we talk about, we are talking about changes which have happened over the last 3,000, maximally 5,000 years. But biology is very much older. But at the very end of my presentation, I must ask a question. Where did the coronavirus come from? Do we know natural evolution, or was it a product of laboratory creation? Because today, techniques exist in virology labs to create synthetic viruses, to do what one might call genetic engineering on viruses. These have existed for some time. For example, the influenza virus causing the 1918 pandemic was reconstructed in the laboratory. But most viruses come by natural evolution. So where did the present coronavirus come from? The answer is we do not really know. I think today the world is divided. Many people favor natural evolution. Many other people favor laboratory creation or laboratory leak at least. Therefore, this debate will go on for some time. But when we ask this question about the coronavirus, we can go back now to the old literature. In the 18th century, the English poet William Blake looked at two biological organisms, the lamb and the tiger. Both of them, when you look at them, and they're pictured on the slide, they show you that marvelous bilateral symmetry. The lamb, of course, looks beautiful. The tiger looks beautiful, but it's also fearsome. What of the coronavirus? Spherical, spikes nicely distributed on the surface. It does look beautiful, but it is also a fearsome object today. Blake asks a very interesting question in his poem, The Tiger. He says, what immortal hand or eye could frame thy fearful symmetry? Did he smile his work to see? Did he who made the lamb make thee? So he asks a question there. That's the kind of question we're asking even today about the coronavirus. Why would we imagine that the coronavirus might be a laboratory creation? This is largely because people have suspicions. Why do they have suspicions? Because the world is a dangerous place. In thinking about suspicion, I came across these quotations from an author who wrote wonderful spy novels a long time ago, not the kind of spy novels that people like to read nowadays, John Le Carre. He says survival is an infinite capacity for suspicion. But he goes on also to say, a desk is a dangerous place from which to watch the world. And over the last year and a half, all that I've done is sit at my desk and try to see what's going on in the world from my desk. And in reading about the coronavirus, many questions do arise. I will conclude my presentation at this point, first by thanking you for inviting me, and then by thanking the two institutions which have provided me a home for much of my professional life. The Indian Institute of Science on the top left, where I spent 42 years, and the National Center for Biological Sciences at the bottom right, which has provided me a place of shelter in my retirement. Thank you very much.
Thank you very much, sir, for such an wonderful and insightful talk. And I was feeling like that this biology and chemistry, this bridge is kind of is a philosophical things. We may think the philosophy behind this two subject that are working hand by hand. And as you showed, Sapiens is one of my favorite book. I enjoyed that book a lot and learned a ton from that. And thank you for such a wonderful talk where we really enjoyed the bits. So now the session is open for questions answer. So please write your questions on the chat sections, and you may write your questions to the comment section of YouTube. So we already have quite a few questions, okay, so Dr. Kamakshi Shurekha asked you that, thank you, sir, for such an illuminating talk. How can we prevent the third wave of COVID-19?
I'm not sure that my answer will be right, because nobody, if we knew how to prevent the third wave of COVID-19, we would be doing it. And so would everybody else. I think now, from whatever I have read, and this is only completely amateurish, I'm not a professional virologist. If you look at the sequences of the variants which are spreading now, the Delta variant, which is the most widely spread mutant, the second phase of the pandemic was caused by the Delta variant in India. That is now in the United States, and it's the driver of the pandemic in the United States right now. One can ask, can the virus play more tricks? I suspect that the virus isn't going to mutate to improve receptor binding beyond what it has done. It's certainly, we are not going to get more effective furin cleavage than the mutations which have taken place. But what I think one needs to guard against is the so-called immune escape mutants. I think genomic surveillance is very important, because what will happen is that mutations happen randomly. And if any of those mutations which happen randomly are now going to be resistant to the antibodies which we have, those will begin to multiply and slowly take over. But I suspect what will happen is what has happened in past viral pandemics. From a pandemic, we will go to an endemic stage where the virus remains circulating in human populations, but does not cause as much damage as it has done so far. At that point, we coexist with the virus.
The next question is from Shomodip Goswami. He wrote, thank you, Professor Balaram, for the wonderful and insightful talk. Sir, my question is about the viral mimicry, as you also highlighted the furin cleavage site in SARS-CoV-2. These type of cleavage sites are also found in various growth factors in humans. So I was wondering, how does the virus, which are said to be non-living outside a host, co-evolved so beautifully and cleverly to mimic the human interactomes?
No, I don't think this has anything to do with living and non-living. This is a protein sequences also evolve simply by mutation. The cleavage site is the same kind of cleavage site that you will find for proteolytic sites across biochemistry. For example, polyprotein processing happens by such proteolytic cleavage. There are lots of enzymes that we have in our body which are called prohormone convertases, which recognize, for example, dibasic sites, tribasic sites, and so forth. That's exactly what the furin cleavage site is. It could just be something that's been selected out because of the fact that only those viruses which undergo effective cleavage are become more infective. It's been known for a long time in virology that in the lab, when you're trying to grow viruses, influenza viruses, for example, if you add a little bit of trypsin to the virus, then you get more infection because that cleavage is important.
Yes. So, sir, next is from Manisha Bose. Sir, can you please explain the difference between mutation and evaluation once more?
Yes, I can explain it many times if you want, but let me try. See, if biological information now is coded in a sequence of letters, and if you change one of the letters, which is a mutation, that happens randomly. There are many times mistakes are made when the string of letters is copied, for instance. That mistake, if it now turns out to confer a new property on the sequence, which helps it in the environment where it is, it gets selected. Those variants which are not useful just fall by the wayside. So Darwinian evolution at molecular level takes place by selecting those molecules which serve a particular function. Selection for phenotype depending upon what the pressures are. Now, for example, if we take the example of the virus, if through vaccines and antibodies one brings the virus under control and the virus is now no longer able to infect, those mutants which evade the antibodies, what we call immune escape mutants, they would be the ones which would start multiplying. That's evolution. It's not purposeful. There is nothing purposeful here.
Sir, the next is from Padmashri Ghosh. She said, very informative session. Thank you very much, sir. Next is from Sweta Shutradhar. Thank you very much, sir, for wonderful and informative session. From Shanaz Ali, wonderful and informative presentation. Thank you, sir. Kushi Jain, she wrote, sir, are there any chances that the mutation in COVID virus decrease its virulency?
Yes, the virus will settle down eventually because most viruses have in the past. We will coexist with them. After all, this is what we have been doing for a very long time. Most pandemics have died down after some time and then become endemic in the population. So this will, as long as the virus doesn't want to kill you, no, the virus would simply like to multiply. And that's what's happening. If viruses are able to multiply well, they will multiply. And if we now adjust to them, we will also live. See, natural selection is operating here. That's what you must understand. We don't like it now, but suppose the same pandemic had come when we had no medicines, when we had no vaccines, and we had no science and technology. What would have happened? Whatever happened in the 1918 pandemic?
Yes. Sir, Shondeep Paul, he wrote, thank you for this excellent presentation. Whether the concept of supreme power is strengthened or weakened due to this pandemic, will there be any drastic changes in our social structure?
Well, I'll answer your last question. There will never be a drastic change in our social structure because once the pandemic is over, we will just go back to, we've already started going back to whatever we were doing. The idea of whether people will become believers or not, those who want to become will, and those who are not will not. But if you study biology, one of the things in biology is if you start at the beginning and look at the evolution of life, it sort of mitigates against supreme powers.
Sir, from Shomodipta Shamanto. Thank you, sir, for this wonderful webinar session. There is a rumor that says that the SARS-CoV-2 came out from Wuhan of China. What are your views on that?
No, I don't think there's a rumor that the SARS-CoV-2 came from Wuhan. The first SARS-CoV-2 sequence came from Wuhan. The earliest cases were diagnosed in Wuhan. Therefore, one believes that the pandemic originated in Wuhan. The debate is whether the pandemic originated naturally or there was work going on viruses and there was a laboratory leak. That is the, if you ask me what my views on it, my views are unimportant, largely because there is no evidence either way. You can look at the evidence and go one way or the other, whatever is available. There is no definitive evidence at the moment.
Sir, from Shubhankar Sinha, other than the few emergency purpose vaccine as recently developed, what about the current status of development of antiviral drug specific against coronavirus? How long it will take to come to the market?
The antiviral drugs have been generally very difficult. In fact, there's success story is what happened in the case of HIV. But by and large, antivirals have been a major challenge for pharmaceutical research as compared to other kinds of pharmaceuticals, antibacterials, other kinds of things. So I don't believe that an effective antiviral is going to appear on the market very quickly. No. Vaccines are a different matter. They have already come and they will continue to come.
So from Shuja Chattopadhyay, sir, what do you think about the possibility of mutational meltdown, where an excess of deleterious mutations accumulate over time, too much to get purged out to the population, thereby making the virus less virulent over time and coexisting with other flu viruses?
Yes, the virus will, see, eventually we come to equilibrium with viruses. We've encountered viruses over the entire course of human existence. And today we consider all this because we are in an age where information transfer is very rapid, where travel is very rapid. So the pandemic has gone all over the world very quickly. This would not have happened 200 years ago. The pandemic would have been more localized. So eventually this will happen. That mutant strains which multiply well, but maybe don't cause, see, much of the damage which is done even today is because of host immune response. A very vigorous host immune response sometimes does lead to problems also.
From Kaushik Vishal, always been a privilege to listen to Professor Balaram right from my PhD days. What an amazing way to visualize chemistry and biology with respect to coronavirus. Retarding the furin cleavage size, recently Sir David Baltimore pointed out the codons of the arginine residues as the furin cleavage site. That this codon usage is something which is not available in other virus or in humans. Does it not point towards an artificial designed virus?
Yes, I might tell Kaushik this, that I did write an article on what Baltimore had said in Current Science some time ago, I think in the month of June. And because of Baltimore's statement, I studied the arginine codons. I don't think that is definitive evidence. In fact, Baltimore himself now says that he didn't mean it that way, but it is unusual. It's not definitive evidence that it's an engineered virus. That's all I could say. But it certainly raises a doubt.
Sir, from Khushi Jain. Sir, do you think epigenetics has played any role in evolution and natural selection?
So this question, I don't think I can quite answer because I am not completely familiar with everything that is attributed to epigenetics. But I can't say that modifications which have taken place uh, afterwards don't have an effect. They might. Because in order to do further modifications, you need other biochemical transformations, and therefore those might be selected over a period of time. So it is possible. I'm sure there are people looking at this, but I'm not familiar with this area.
Okay. So no more questions. I can't see anything more. So one very different questions from my side, sir. So what we know, disease-related proteins has some percentage of intrinsically disordered region in it. Say cancer-related proteins or the Alzheimer-related proteins, Parkinson's, etc. But for this kind of virus, the virus proteins are mostly ordered in structure. Can you tell me that why the virus-related proteins, preferred more ordered structure than the disordered?
Yeah, first of all, let me say one thing that if you look carefully at the coronavirus spike protein, if you take 1273 residues, there are two distinct regions, the first 600 and the second 600. So the N-terminus 600 contains a large number of segments which are actually disordered. It's not very structured. The receptor binding domain, the N-terminus domain, the furin cleavage site, these are all disordered regions of the protein. Considerable disorder there. Many of the key mutations which have led to variants are taking place in those regions. So, but the second part, the C-terminus domain, is very much more structured because you need those helices in order to come together to do that membrane fusion. So both the spike protein actually gives you a wonderful example that you can discuss both order and disorder at the same time.
Okay, sir. Uh, No more questions, but all the thank you and wonderful presentations, a lots of messages from the participants. And so thank you very much, sir, for accepting and for this wonderful sessions that we have together. My sincere gratitude from the behalf of our JISIASR family, and thank you very much. So now we can
Thank you.
end the session. So for all the participants, so you can leave
Thank you,
that.
Devamni.
Thank you, sir. Thank you very much.